synaptic vesicles. This is a membrane covering the muscle called sarcolemma. Here we will get some receptors. Or you will get some receptors, they are called the receptor. This part is sarcoplasm. Clear? This is called the neuromuscular junction. Neuromuscular junction. Junction between nerve and the muscle. Now what happens here? To initiate muscular contraction, the information or impulse is required to be carried to the muscle from the nerves. Otherwise, the muscular contraction cannot be initiated. So what happens here actually, whenever impulse comes to the nerve endings, the synaptic vesicles are found in the ending part of the nerve, end part of the nerve. These vesicles are filled with neurotransmitters. Neurotransmitters like acetylcholine or adrenaline, whatever it may be. Now, whenever impulse comes to this, basically the membrane, this is called the presynaptic membrane. This is the presynaptic membrane, and normally this membrane is not allowing calcium ion to enter inside. But whenever the impulse comes to the end part of this nerve, it causes the change in the permeability of the presynaptic membrane, so that from the intercellular space, calcium ion enters and stimulates this vesicle. I am repeating it again. Normally the nerve endings, in the nerve endings you will get some, or at the junction of the nerve and muscle in the nerve side, there are some synaptic vesicles filled with neurotransmitters. Then these neurotransmitters are to carry transmission and impulse from nerve to muscle. But for this, these neurotransmitters are required to be stimulated. Now the question comes, who is to stimulate this neurotransmitter? Only calcium ion can stimulate neurotransmitters as the synaptic vesicles to release neurotransmitter. Normally calcium ion is found in the extracellular fluid between the nerve space, between the nerve and muscle, but the membrane of the nerve, that is presynaptic membrane, is not allowing calcium ion to enter the synapse area or synaptic knob. Then what happens? As soon as the impulse comes, the permeability of the presynaptic membrane changes, so it is found permeable to calcium ion, or calcium ion channels open, and from the extracellular fluid, calcium ion enters the synaptic vesicles. After it is entering into the synaptic vesicle, it stimulates the synaptic vesicles to release acetylcholine or neurotransmitter. Then this neurotransmitter then diffuse out and binds to a particular receptor present over here, and again the permeability of the sarcolemma changes. Now from the extracellular fluid, sodium ion enters, so that the gradient is developed, concentration of sodium ion increases, and which causes the development of a gradient. And in this way, once gradient is developed, means impulse is carried to the muscle side, so the neuromuscular junction. I am repeating it again. Listen carefully. Generally, in the synaptic knob, this is the synaptic knob, you see. Here you will get a number of vesicles. So, this is the synaptic vesicles. You know, and so this is the sarcolemma. And here you will get the extracellular fluid. And different ions are present here. So these synaptic vesicles are filled with neurotransmitters, which are required to carry impulse from nerve side to the muscle side. So, but you know that to release neurotransmitters, synaptic vesicles are required to be activated. Only calcium ion can activate them. But the membrane of the synaptic vesicles, this membrane called presynaptic membrane, it is not allowing calcium ion to enter inside. But whenever impulse comes, then what will happen? The permeability of the membrane changes. Now it is allowing calcium ion to enter inside. This will stimulate these vesicles. Comes in contact with, then what happens? There comes in contact with the membrane and neurotransmitter is released. This neurotransmitter in turn will bind to the receptor. This is the receptor. Neurotransmitter binds. As soon as neurotransmitter binds to the receptor, permeability of the sarcolemma changes. Now sodium ion enters. So that gradually a gradient is developed. And once the gradient is developed, you can say that nerve impulse is carried from nerve side to the muscle side. Is it clear? How impulse is propagated from nerve side to the muscle side through neuromuscular junction?
Sir?
Bolo.
The calcium ion কিভাবে enter করে protect করব?
Calcium actually in the first of all you need to understand one thing. In the extracellular fluid, different types of ions are there. Every ion has a particular channel in the membrane. Until this channel is open, the ion cannot enter inside. Are you getting me this?
Yes, sir.
Now, generally, normally in resting stage, when impulse is not coming to the nerve ending, the membrane or presynaptic membrane, in the presynaptic membrane, the calcium ion channels are found to be in closed form. Clear?
Yes, sir.
Although calcium ions are in the extracellular fluid, but these calcium ions cannot enter inside. Okay?
Okay, sir.
Then what happens? After that, you see, these are the membranes. Whenever impulse comes to the nerve ending, this impulse changes the permeability of the presynaptic membrane. Now, calcium ion channels are found to become open. Clear?
Okay, sir.
Now, through this channel, calcium ion enters inside the synaptic knob, stimulates these vesicles so that they come in contact with the membrane and the secretion is diffused out. Clear?
Yes, sir.
This secretion, that is acetylcholine, will bind to the receptor present in the sarcolemma. Once neurotransmitter binds to receptor, the permeability of the sarcolemma changes. Now from extracellular fluids, sodium ion enters the sarcoplasm and gradually its concentration increases so that a gradient or difference in concentration developed, and this is called action potential, or you can call it nerve impulse. In this way, nerve impulse is carried to the muscle side. Once impulse is carried to the muscle side, this impulse is to stimulate sarcoplasmic reticulum. Action potential
স্যার সোডিয়াম যে বাইরে থেকে ভেতরে যায় এটা একটু বলে দেবে?
বলছি তো। একটু action potential stimulates sarcoplasmic reticulum. Then from this calcium ions are released, carried here and binds to the troponin to make the troponin inactivated. So wherever, as per the impulse, contraction occurs, and so what happens here, whenever calcium ion enters, is released from the sarcoplasmic reticulum, they connect to the site of muscular contraction and bind to troponin. Troponin gets inactivated. Once troponin gets inactivated, tropomyosin sheet moves away. Tropomyosin sheet moves away. Once tropomyosin sheet moves away, what happens? The myosin side, binding site of actin or active sites of actin are exposed now. That is, at this point, the myosin head can bind to actin. Are you getting me? Up to this? Doubt থাকলে বলো। Please.
স্যার সোডিয়ামের জায়গাটা থেকে আরেকবার বলে দাও।
Sodium ions are found in the extracellular fluid. Whenever acetylcholine released out from the synaptic vesicles binds to the receptor present in the sarcolemma, the permeability of the sarcolemma changes so that the sodium ion channels present in the sarcolemma remain open and through which sodium ion enters the sarcoplasm, and a action potential is developed due to the development of a gradient which is called nerve impulse. This action potential of nerve impulse in turn will stimulate sarcoplasmic reticulum. From sarcoplasmic reticulum, then calcium ions are released. Calcium ions will then carry to the site of contraction and binds to troponin. Once calcium ion binds troponin, then it gets inactivated. Once troponin gets inactivated, tropomyosin sheet moves away so that the active sites of actin are exposed now. Once active sites of actin are exposed, now myosin head can bind to actin. That is, muscular contraction can be initiated. দেখো অবধি দেখো। ডাউট থাকলে বল।
সার্কোপ্লাজমিক রেটিকুলামের মধ্যে কি ক্যালসিয়াম আয়ন স্টোর থাকে?
হ্যাঁ। The storehouse of calcium ion. And once muscular contraction is completed, then these calcium ions are going to return to the sarcoplasmic reticulum by calcium pump. দেখ কোন ডাউট আছে কিনা।
স্যার ক্যালসিয়াম গুলো কি রিসাইকেল হয় নাকি?
হ্যাঁ calcium
নতুন
pump,
ক্যালসিয়াম লাগে না?
calcium pump, whenever muscular contraction is completed, then these calciums are required to be removed out, otherwise the muscle cannot be at resting stage. Because once contraction is completed, troponin is required to be active again. For this, calcium ions are to be released. So by calcium pump, then calcium ions are released and return to the what? Sarcoplasmic reticulum. Clear? A of the?
Yes,
Is clear up to this?
স্যার সোডিয়ামটা কিভাবে অ্যাকশন পোটেনশিয়াল জায়গাটা?
When the concentration of sodium ion increases, as regular entry of sodium ion causes higher concentration of sodium ion at one point here, so that there is a difference in gradient, and this difference in gradient is called the action potential or impulse. Impulse is nothing but the difference in concentration gradient of sodium ion. Clear?
Yes sir, yes sir.
Now, is it clear up to this part?
Yes, sir.
Okay.
Yes,
Now,
sir.
I will rub it out. Can I rub it out now?
Yes, sir.
Calcium ion binds to troponin and troponin gets inactivated. Tropomyosin sheet moves away. Calcium ion binds to troponin. Troponin gets inactivated. Tropomyosin sheet moves away. Active sites of actin are exposed. Now, myosin head is bind to the myosin head binds to actin. Clear? Before that, before binding with this, what happens here actually, the myosin head, want to shape one diagram. What happens here, I am showing you here first. So what happens here actually, ATP binds to it, ATP binds to it, hydrolysis of ATP, so that the energy produced, look one thing, it bends like this way. ADP, and P. So it is broken down to ADP and P. And it is actually pivoted or binds. It moves to this direction. That means it was like this. Now it is shifted like this. Clear? Pivoted. Once it's pivoted, so in this diagram we can draw this like this. The pivoted ADP, and you will get the P. It's bent due to the energy it pivots, and then what happens after that? Next diagram. Now, myosin head binds to actin and a cross bridge is found here. So here it is. Cross bridge is formed. Once myosin head binds to actin, the cross bridge is formed. But you see, ATP is already exhausted. As ATP is already exhausted, the myosin head tries to return to its original position. So it is found to pull the actin along with it, because so here what happens once myosin head binds to actin, actomyosin complex is formed. So this is what happens here. Actomyosin complex is found here. But as ATP is already exhausted, so the myosin head tries to return to its original position. देखो ये सब बुझते हो कि ना? এই অবধি দেখো আগে বুঝো কিনা I said earlier, the head of the myosin is actually an active ATPase enzyme. ATPase enzyme is an enzyme that causes hydrolysis of ATP. So in the myosin head there is a site for the binding of ATP. So whenever ATP binds to it, so ATPase causes hydrolysis. So then ADP and PI produced along with the production of some amount of energy, so that the head is pivoted, actually aligned. Look, one thing is the original one. I am showing you this one, and that is aligned to some extent. And after alignment, then what happens? Then the head is going to bind to the actin protein. Once it binds to actin protein, then actomyosin complex is formed. Is it clear?
Yes, sir.
স্যার এডিপি আর পি ফর্ম করার পর এটা পাইভট হয়ে গেছে।
And then it binds. It pivots and binds. And again, whenever you see, for binding with the myosin, the head is required to be pivoted, and this is facilitated by the hydrolysis of ATP. Then it binds. Once bind, then it tries to return to the original position because already ATP is exhausted. But already you know, to facilitate actomyosin complex formation or to help in the breakdown of this complex, in both the cases ATP is required. In both the cases ATP is required. So ATP is required in both, and as you see, as ATP is required in both the cases, so what is most important here? So until ATP is available, actomyosin complex cannot be broken down. So while myosin head is trying to return to its original position, it is to pull the actin along with it. It is to pull the actin along with it. So what happens, I am showing you now in the next diagram. So what happens? This is the troponin protein. Calcium ion attached to it. Then what happens? What I said, it is trying to pulling. So it rotates actually like this. 1, 2, 3, 4, 5. The fifth one. So it is called rotation. After rotation, at this point, suppose ADP and PI comes. After that, so what happens next, you see? So it is detached. The head gets detached. Whenever again ADP and PI available, head gets detached. Now this head, in the meantime, it proceeds toward the center. So now the head gets 1, 2, 3, 4. These actin in front of it. Because it proceeds toward this, because it is pulling, so it proceeds toward the center. So this head of myosin now get a new actin in front of it. Head of myosin will get a new actin in front of it. Are you getting me now?
Yes, sir.
New actin in front of it, and again it binds to it similarly. It binds to it similarly like this. and eventually the process is going on. Clear? It is pivoted in the form of pivotation. It is, you can draw it like this, pivoted. Okay.
স্যার
Yes.
স্যার রোটেশনটা হয় কেন?
Look, one thing, what I said, one most important thing is that the myosin head binds to actin. This is true. But whenever ATP is exhausted, then myosin head that binds to actin tries to return to its original position, right?
Yes, sir.
Then it is pulling, but you know, for the formation of ectomyosin complex and also for the breakdown of ectomyosin complex, ATP is required, right?
Yes, sir.
So if ATP is not available, then if it is to return to original position, it is to bring the myosin actin along with it.
Yes, sir.
And as it is actually pulling, in this way it is pulling the actin.
Yes, sir.
As this is the causal rotation. Clear?
Okay, sir.
স্যার লাস্টের ডায়াগ্রামটাতে ট্রপোনিন থাকবে না?
হ্যাঁ, থাকবে। Sorry. So the process continues till the contraction is completed. And this is the rotation actually. Pulling. If you go to this diagram, then it is easier for you to understand. So suppose this is the Z line, Z. So here this one, so this is the actin. Due to the pulling effect, what happens gradually, Z lines are found closer. If I am going to show the same diagram, after some time you will get this Z. And if it is so, then you will get like this. That means that length between the Z line decreases. The length of sarcomere decreases as whenever the myosin head is pulling the actin, actin is proceeding towards the center. When myosin is pulling the actin, actin is gradually proceeding towards the center. And one time will come whenever it appears like this, there is no existence of H line. Then you can call it the muscle is contracted. Are you getting me? Every time when it binds, again pulling occurs, again bind, again pulling occurs, the process is repeated. So that gradually the length of sarcomere decreases. And as length of sarcomere decreases, eventually there will be no existence of H line here. And you can call it one is overlapping the other, which may be termed as M line. And then you can call it muscular contraction is completed. Is it clear to you? Because the process is repeated continuously till the M line is formed. Once muscle is fully contracted, then further shortening will not occur. Then for relaxation, calcium ions are required to be released out from the troponin and again muscle return to its original position. দেখো বুঝতে পেরেছি কিনা?
স্যার লাস্ট ডায়াগ্রামটাতে এই লাইনটা একটাকে একটা ওভারল্যাপ করেছে কেন?
হ্যাঁ তো actually as actin is gradually pulled by the myosin so that it moves towards the center. Whenever actin is pulled by myosin, due to this pulling, actin is gradually proceeding towards the center and so that from both sides, if it proceeds towards the center, obviously one is found to overlap the others.
Yes, sir.
This is clear? দেখো ভালো দেখো না। Always, whenever myosin head is pulling the what is called actin, gradually it pulls towards the center of the event. And that's why the length of sarcomere gradually decreases. This is the cause why length of sarcomere gradually decreases. Is it clear? I am repeating it again. When calcium ion is released from the nerve endings, it comes actually, calcium ions are released with the sarcoplasmic reticulum. After the nerve impulse comes to the sarcoplasm, this calcium ion binds to troponin. Troponin gets inactivated so that tropomyosin sheath moves away and active sites of actin are exposed now. Now, in the myosin head, hydrolysis of ATP occurs, so that it moves away, it actually pivoted and binds to a particular actin protein. So that actomyosin complex is formed. Once actomyosin complex is formed, ATP is exhausted. So that myosin is to return to the original position, so it starts rotating. While it's actually starts rotating the myosin, the actin is proceeding towards the center of the event, and in the meantime, this is called rotation. And in the meantime, ADP and PA are released out. So the new ATP comes, and bind to it, and again the same thing happens. ATP and PA is formed, and again the contraction going on, and this so that gradually length of sarcomere decreases, and once contraction is completed, then by calcium pump, the calcium ion is required to be released out from the troponin and return to the sarcoplasmic reticulum. Once troponin is free of calcium ion, it becomes active, and then again tropomyosin sheath is found to cover the active sites of actin, and the process of contraction actually completed. Now it is in the resting stage. Whenever myosin binds to actin, this is called cross bridge. So again, cross bridge is broken down when ATP is available. Because whenever ATP is available, ATP is converted to ATP and PI. So cross bridge is broken down. Once cross bridge is broken down, again, what happens? Again, new cross bridge is formed, and the process continues. So that gradually the length of sarcomere decreases, although the event remains same, but length of event also reduced. Event gets reduced, but event maintains its own length. Length of event remains same. দেখো এটা বুঝতে পেরেছি কিনা? ডাউট থাকলে বলো। দেখে নাও ভালো করে।
স্যার সেকেন্ড স্টেপে তো এটিপিটা এক্সস্টেড হয়ে গেছে, তারপরে আবার থার্ড স্টেপে এডিপি আর ফসফেট কি করে প্রডিউস হচ্ছে?
প্রডিউস হচ্ছে না তো, এই যে এটিপিটা তো এডিপি আর পিআই, যে এটিপি যখন এক্সস্ট হয়েছে কি বেরিয়েছে, এডিপি আর পিআই আছে না, এই যে এগুলো বেরিয়ে গেছে, এখানে আর দেখানো হয়নি।
আচ্ছা স্যার,
When ATP get hydrolyzed, what is produced?
ADP and phosphate.
ADP and phosphate. And these ADP and phosphate are here. After rotation, they are released. Then
Yes,
again
sir.
ATP binds. Again hydrolysis occurs. Then
Yes,
বুঝতে
sir.
পেরেছি।
হ্যাঁ স্যার, এবার বুঝেছি।
ক্লিয়ার? মনে আছে কি মায়োসিন একটি লেখা আছে তো একটা মিনিট। হ্যালো to everybody.
Yes, sir.
Now, however, repeated contraction, skeletal muscles, in case of skeletal muscle, refractory period is short. So repeated contraction can occur as per requirement. Then it leads to the formation of lactic acid. Leads to clear? If Z গুলি কি? Okay, সহি নেই। Z actually, Z line, can't you remember Z lines? Okay. In a myofibril, this is the event. Again event. So, in case of myofibril, event and I-band occur alternately, or dark band and light band. So this is the dark band. And this is the I-band. Called is light band. Dark band and light band occur alternately. In the center of the event, the lighter line is called H line. And in the center of the I-band, the darker line is found called Z line. Is also Z line. And distance between to Z line is called sarcomere. So the center of the I-band, actually A-band and I-band are alternately found in the myofibril. In case of skeletal muscle, you will get alternate arrangement of A-band and I-band. A-band refers to the anisotropic band, or you can call it dark band, and I-band refers to the isotropic band, or you can call it light band. In the center of the A-band, there is a lighter line called IH line. In the center of the I-band, there is a darker line called Z line. And distance between two successive Z line is called sarcomere. So this is the distance between two successive Z line is the sarcomere, which is called unit of myofibril, or sometimes called unit of muscular contraction. Is it clear, Shainim? Okay. So real arm H line, I'm rubbing it out, please.
স্যার
Yeah.
স্যার এইচ লাইন থেকে এইচ লাইনকে কেন একটা সার্কোমিয়ার ধরে না?
What is sarcomere? Consists of a complete event and half of each event. কথা বুঝতে পেরেছো? What are written? Sarcomere consists of a complete event and half of each event.
ইয়েস স্যার।
H line is also called Hansen's line, and Z line is called Krause's line. You can call it Krause's line or Dobie's line. Krause membranes. Or Dobie's line. Try to remember this. So this is the H line. This is the H line, also called H line. Is it clear? The how is what is called? Due to repeated contraction, lactic acid is found to be accumulated in the muscle and muscle get fatigued. Question is, how is lactic acid accumulated? How is lactic acid formed? Can anyone tell me? How is lactic?
So when ATP is finished, so to maintain, so for the requirement,
yes.
for the requirement of ATP, muscle starts doing anaerobic respiration,
Then
and
what
then
happens?
lactic acid is formed.
No. Actually, muscle, from which compound lactic acid is formed? Actually, what is going on here?
Sir, pyruvic acid.
Actually, first of all, whenever energy demand is developed, there is a demand of energy in the muscle. Whenever there is demand of energy in the muscle, then what happens, I am telling you. Whenever there is demand of energy in the muscle, glycogen stored in the muscle undergo glycogenolysis to form glucose. Clear? Glucose then glycolysis to form pyruvic acid. Pyruvic acid then to form lactic acid plus energy. Almost 36 kilocalorie energy is produced, and this is required to meet the energy demand at that time. So in this way, if continuously lactic acid is accumulated, then the muscles become fatigue. It cannot contract, so we feel pain. I think I discussed earlier why cardiac muscle never get fatigue. Can you remember? Onirban. Why
Yes, sir.
cardiac muscle never get fatigued?
স্যার রিজন গুলো বলবো
হ্যাঁ,
ফার্স্ট তো there is more myoglobulin present in cardiac
myoglobin, not globulin. হ্যাঁ।
then LDH1 in cardiac tissue
Which is prevented by or which is inhibited by?
pyruvate
Very good. And third one.
refractory period of cardiac muscle is long.
Very good, very good, very good. There are three basic causes why cardiac muscles never get fatigued. One is the amount of myoglobin is much more, which acts as oxygen reservoir and also supplies oxygen to the tissues. Secondly, the refractory period is long, as there is long refractory period, so the muscle has time to relax. And thirdly, lactic acid cannot be accumulated because the enzyme lactate dehydrogenase is found in LDH1 form, and which is actually whenever any enzyme is found to be in LDH1 form means it is inhibited by the substrate pyruvate, hence lactic acid cannot be developed in the cardiac muscle. So how, but you know, in the skeletal muscle, lactic acid is required to be developed to meet the emergency, because you know, whenever we are exercising or whenever we are running, obviously lactic acid is required to be formed to meet the energy demand at that very period. If lactic acid cannot be formed in the skeletal muscle, then it would be dangerous for us, but it is not possible for us to survive again. That's why it is an adaptive feature that in the skeletal muscle, LDH5 is present to facilitate lactic acid formation, but in the cardiac muscle, lactic acid, as it is not required to be formed, that's why LDH1 is present. Clear to everybody?
Yes, sir.
Yes, sir.
The cause of muscular fatigue is clear, and second, similarly, it is also clear that how can we be relieved from the pain? Whenever we take rest for a while, then what happens? The lactic acid again converted to glycogen via water. Lactic acid is to form pyruvic acid, then it is to form the glycogen and stored in the muscle. So this is what about the muscle fatigue and its consequences. Clear?
Sir,
Bolo.
sir, if you give hot water on the area, on the muscle, hot water.
Basically, one thing I am telling you is a pain reliever. Actually, one thing I am telling you, that lactic acid is required to be removed out from the muscle. For this, the most important requirement is oxygen. Because in the presence of oxygen, NADH, because in the reaction you see what is required. Go to the reaction. Lactic acid look on. This is the reaction. That means NAD plus. This is the oxidized form. This is the reduced form. NADH means reduced form. NAD plus is the oxidized form, NADH is the reduced form. So NAD should be present in oxidized form to facilitate conversion of lactic acid to pyruvic acid. So how can it be possible? Because NADH can only be converted to oxidized form in the presence of oxygen. That's why in the oil ventilated room, it is required to rest in oil ventilated room for facilitating conversion of lactic acid to pyruvic acid. Are you getting me?
Yes, sir.
So this is all about the, now here's something more here regarding the different types of muscles. Actually, regarding there are two types of muscles. You should go through it. If possible, tomorrow, the next day, I'll be showing you one video to make you understand the muscular contraction again. ni positivo like I think I have one. I can't trace it now. Okay. So this is what about the muscular contraction, and here also we have to discuss.
Sir?
Yes.
স্যার ল্যাকটিক অ্যাসিড কি করে যে মাসেল ফ্যাটিক হয়ে যায়?
You know, whenever lactic acid is accumulated, how muscles can contract? Myosin cannot bind to actin. Space is accumulated by lactic acid in the muscle. Until lactic acid is removed out, the myosin cannot bind to actin to initiate muscular contraction. বুঝতে পেরেছো?
Yes, sir.
Today I am going to close now. Tomorrow, then the next Wednesday, actually, your script is actually, তোমাদের খাতা তো দিন না? মার্কস তো জানো না বোধহয় এখনো?
No, sir.
হ্যাঁ, দেখ আজকে দেখে দেবে। আর নেক্সট দিন আগে তোমার পেয়ে যাবে এটা। আর নেক্সট
Sir,
দিন একটা পরীক্ষা হবে কিন্তু।
স্যার র্যাঙ্কটা দাও নেই এখনো।
ও, ওই যে আগের র্যাঙ্কটা। সবাই পেয়েছে তো রেজাল্ট? যেটা মানে 160 মধ্যে নিয়েছিলাম পরীক্ষার বোধহয়।
ইয়েস স্যার।
নেক্সট দিন কিন্তু পরীক্ষা হবে আবার। নেক্সট দিন কিন্তু আজকে যেটা পড়েছি আর নেক্সট দিন যেটা পড়াবো তার উপর পরীক্ষা হয়ে যাবে। ক্লিয়ার?
ওকে স্যার।
ঠিক আছে।
আমাদের কিন্তু একবারে কিন্তু মুভমেন্ট ও লোকোমোশনের মাসলটা পড়া হয়ে যাচ্ছে, এটা শেষ হয়ে যাবে কিন্তু, এটাও প্রায় শেষ হয়ে গেছে। দুটো জিনিস আমি পড়াইনি আজকে, এটা আজকে আর মানে পড়ার সময় হয়ে যাচ্ছে। রেড মাসল ফাইবার, হোয়াইট মাসল ফাইবার এন্ড দ্যার ইম্পর্টেন্স। ক্লিয়ার?
হ্যাঁ স্যার, ঠিক আছে।
ওকে,
Thank you, sir.
ওকে, থ্যাংক ইউ।